I'm Joseph Zhao. So I will present about genetic engineering and recombined DNA. Genetic engineering are artificial manipulation, modification, and recombination DNA or other nucleic acid molecules in order to modify in an organism or population of organisms. Genetic engineering is generally referred to the methods of recombined DNA technology, which emerged from basic research in microbial genetics. Recombined DNA is created by combining two different fragments from two different sources. New DNA is obtained by either isolating and copying the genetic material of interest using recombined DNA methods by artificially synthesizing the DNA. Recombined DNA is possible because DNA molecules from all organisms share the same chemical structure. But the difference is only the nucleotide sequence within that identical overall structure. Recombined DNA called chimeric DNA. This is because they can be made of two materials from two different species. Recombined DNA technology use palindromic sequences and lead to the production of sticky and blunt ends. The DNA sequences used in construction of rDNA molecules or can originate from any species. For example, plants DNA may be conjoined to bacterial DNA, or human DNA can join with fungal DNA. DNA sequences that cannot occur anywhere in nature may be created by chemical synthesis of DNA and incorporated in recombined molecules. So next, I will pass this presentation to Enali.
Thank you, Joseph. Cloning. The recent discovery of cloning tools in Streptomyces has enabled the isolation of biosynthetic genes for some of the numerous antibiotics produced by members of the significant genus of bacteria. These clones can now be used to test the theory that new antibiotics could emerge from the exchange of biosynthetic genes between streptomyces that produce different antibiotics. What is antibiotics? Antibiotics are powerful drugs that combat infections and, when taken correctly, can save lives. They either prevent bacteria from multiplying or kill them. Germs are bacteria. They both live in the environment and all on our bodies, both inside and outside. However, there are occasions when the number of dangerous germs is excessive and the immune system has been unable to combat them all. Antibiotics can be handy in this situation. According to Russell, antibiotics are organic substances that produced by one microorganisms which are toxic to another microorganisms. According to Waltz, while some bacteria can completely kill bacteria, others can only stop them from multiplying. Virostatic antibiotics can kill bacteria, whereas bactericidal antibiotics suppress bacterial growth. Antibiotics are classified as antibacterial, antifungals, or antiviruses, depending on which microorganisms they fight. Now, I would like to pass the time to Audrey.
Okay, Bismillahirrahmanirrahim. Assalamualaikum. My name is Muhammad Hakim. I would like to share about plasmid. Okay, a plasmid could be a small extra chromosome DNA atom inside a cell that physically isolate from chromosomal DNA and can be reproduced freely. They are most commonly found as a little circle double standard DNA, particularly in a microscope organism. Be that as it may, plasmid are some of the time displayed in arched and eukaryotic living being. Okay, next, the insert DNA carry the complete cluster for genic of the biosynthesis of actinor and the last is plasmid may be classified in a number of ways. Plasmid can be broadly classified into conjugative plasmid and non-conjugative plasmid. Okay, for conjugative plasmid contain a set of exchange qualities which advance sexual conjugative between distant cell. And then to the complete prepare of conjugative plasmid may be exchanged from one bacterium to another through successfully accounted by a few of the exchange quality. And then for the non-conjugative plasma unfit of starting conjugative, thus they can be exchanged as if were with the help of conjugative plasmid. Thank you.
Hi, my name is Noor binti Sedan and my number matrix is F21A1212. So how genetic engineering can produce antibiotic? The recent development of molecular cloning system in streptomyces has made possible the isolation of biosynthetic gene for some of the many antibiotics produced by members of this important genus of bacteria. It has been demonstrated that antibiotic biosynthesis genes are clustered in a single, usually chromosomal segment. The organization of genes currently identified for the biosynthesis of the macrolide antibiotic erythromycin in Saccharopolyspora reticula. Gene description resulting from insertion of DNA into a coding sequence is technically the simple method to activate genes and has been used for a number of antibiotics. Genetic manipulation of Saccharopolyspora reticula has resulted in the production of 3 novel compounds and the overproduction of an important pathway intermediate. The aromatic polypeptide comprise a large family of antibiotic and anti-tumor agent. Thank you. I will pass to the next presenter.
Thank you, Mazini. I will continue where she left off. Hi, my name is Mohammed Ashfield bin Mohammed Hill. Number matrix is F21A1114. The most commercial important type are the tetracycle and the anthracycle dose-robin. Including in this family are the isochromoquinons, dehydrogenetics, and antinodine. Those biosynthesis genes are the best characteristic for antibiotics. This is because the gene for the synthesis of isochromoquinons, dehydrogenetics, and antinodine have been employed in the first example of biosynthesis of hybrid biotics. Dehydrogenase was purified by the column's chromatograph on acrylic acid treated silica gel, which is chromoforms and preventive of TCl, and was converted to the methanol ester. Meanwhile, antinodine was a acid-base initiator which confer characteristic color on the culture, producing color of red-purple. Next, Audrey will continue with the summaries.
Okay, and for the conclusion, what we learn today, this chapter discuss the application of genetic engineering technology to antibiotic biosynthesis. It has been demonstrated that antibiotic biosynthesis generate culture in a single, usually chromosomal and segment. Next, the example present in the chapter illustrate that the application of genetic engineering technology to antibiotic can result in the production of novel structure and improve titles. Both application depends on detailed knowledge of the biochemistry and genetic of the pathway concept. That's all for today. Thank you to lecturer and thank you for all members. Assalamualaikum warahmatullahi wabarakatuh.